Welcome to today's lecture on histology, specifically focusing on the detailed structure and functions of the nail. Throughout this script, we will delve into various topics such as nail ultrastructure, onychocyte differentiation, embryological origins, nail pathology, immunohistochemistry techniques, and many more. So, let's jump right in. The nail, also known as the nail plate, is a hard, translucent structure located at the distal end of the fingers and toes. It provides protection to the underlying tissues and enhances manual dexterity. The nail is primarily composed of keratinized epithelial cells, known as onychocytes, which are organized in layers. The nail plate can be divided into several regions, including the free edge, the body, and the nail root. The free edge is the part of the nail that extends beyond the digit. The body is the visible portion of the nail, while the nail root lies beneath the skin and is responsible for nail growth. Onychocytes are specialized epithelial cells that undergo a complex process of differentiation and keratinization to form the nail plate. This process involves the proliferation of cells in the nail matrix, located at the nail root. As onychocytes move forward, they undergo various changes, including the synthesis and deposition of keratins, as well as the formation of keratohyalin granules. These granules play a crucial role in the cross-linking of proteins and contribute to the hard and durable nature of the nail. The onychocytes eventually become fully keratinized, forming the nail plate. During embryogenesis, the formation of the nail begins with the interaction of the epidermal and mesenchymal cells in the developing limb bud. The epithelium thickens and forms the nail field, which eventually gives rise to the nail plate. Molecular signaling pathways, such as the WNT and SH signaling pathways, play essential roles in nail development. These pathways regulate the proliferation and differentiation of cells in the nail matrix, ensuring proper nail formation. Onychodystrophies are a group of nail disorders characterized by structural abnormalities in the nail plate. These conditions can arise due to various factors, including genetic mutations, trauma, infections, or underlying systemic diseases. Some common onychodystrophies include nail pitting, koilonychia, spoon-shaped nails, onycholysis, detachment of the nail plate, and onychomycosis, fungal nail infection. Understanding the histopathological changes associated with these conditions is crucial for accurate diagnosis and management. While rare, neoplasms and tumors can affect the nail unit. These can include benign tumors like onychopapilloma or malignant tumors such as subungual melanoma. Histopathological examination and proper diagnosis are essential in distinguishing these nail neoplasms from other conditions and guiding appropriate treatment strategies. Nail pigmentation disorders encompass a broad spectrum of conditions characterized by abnormal coloration of the nail. These can range from localized disorders, such as longitudinal melanonychia, brown or black streaks, to diffuse disorders like leukonychia, white discoloration. Histological evaluation combined with clinical correlation aids in determining the underlying causes of these pigmentation abnormalities, which can include melanocytic activation, vascular alterations, or systemic diseases. Immunohistochemistry, IHC, plays a crucial role in the evaluation of nail pathologies by identifying specific antigens or markers. IHC can help differentiate between various conditions and provide valuable information about cellular processes and molecular alterations. By utilizing specific antibodies and staining techniques, IHC allows us to visualize and interpret the expression pattern of proteins involved in nail development, pathology, and regeneration. Nail phenotyping involves the study of nail morphology and its association with genetic, environmental, and systemic factors. Analyzing factors such as nail shape, lunula size, and texture can provide insights into an individual's overall health and specific diseases. Detailed examination of nail phenotypes can aid in diagnostic decision-making and disease management, making it an essential tool in both clinical and research settings. Nail aging is characterized by various structural and functional changes that occur over time. These changes include increased nail plate fragility, decreased growth rate, and alterations in nail color and texture. Understanding the histological changes associated with nail aging can assist in differentiating between normal aging processes and pathological conditions affecting the nails in elderly individuals. The nails not only serve a protective function but also contribute to the biomechanics of the fingers and toes. The bending and stiffness properties of the nail are crucial for optimal tactile sensation and grip strength. Histological examination combined with biomechanical studies helps unravel the structural components responsible for nail strength, flexibility, and overall functionality. Nail regeneration is a fascinating process that occurs after injury or surgical removal of a portion of the nail plate. The ability of the nail unit to regenerate itself relies on the presence of nail matrix stem cells. Histological studies have provided insights into the regenerative capacity of the nail, highlighting the importance of the nail matrix in restoring the nail plate's normal structure and function. Recent research has emphasized the presence of a diverse microbial community, known as the nail microbiome, on and around the nails. The microbiome plays a crucial role in maintaining a healthy nail environment and protecting against disease-causing pathogens. Histological studies combined with advanced molecular techniques have allowed us to explore the composition and dynamics of the nail microbiome, providing valuable insights into its influence on nail health and disease. In conclusion, the nail is a complex structure made of specialized cells. Understanding its structure, development, and diseases is crucial in dermatology. Today, we discuss the importance of histology and molecular knowledge in studying nails. I hope this overview was helpful. Thank you and see you next time.